Interestingly, photosynthesis accomplishes basically the opposite of what we saw happen in cellular respiration in Chapter 4. In cellular respiration, we saw that a carbohydrate was broken down to produce water and CO2. In the case of photosynthesis, we see that carbon dioxide is combined with water to produce a carbohydrate, glucose specifically, and oxygen. So photosynthesis is carried out primarily by plants and some bacteria. Many bacteria, referred to as cyanobacteria, that are aquatic primarily, are photosynthetic bacteria. But photosynthesis is not carried out by animals or fungi. Carbon dioxide used in photosynthesis enters plants through openings called stomata. So remember, we're going to talk about photosynthesis in just a moment. But for photosynthesis to occur, plants have to have this continual supply of carbon dioxide. And so how do they get that? And the answer is it enters through little tiny holes in the leaves referred to as stomata. On this particular slide we see some pictures of leaves, the surface of leaves underneath the microscope, and they're magnified to the point where we can actually see the stomata. And so what I'm pointing to here and there are the stomata of these plants. And so interestingly, let's go over to this diagram. And so here we see a stomata that's opening. The stomata is the hole in the center here, and on either side we have one here and one here cells. These are two cells that are referred to as guard cells. And so fascinatingly, all plants have this. They all have stomata shown here, and they all have guard cells that surround the stomata. Over here, if you look at this other diagram, you can see that the stomata are closed, and so primarily you just see the guard cells and the little area between there that is the stomata, but the stomata is currently closed. So fascinatingly, all plants face this dilemma. They have to keep these stomata open in order for CO2 to move into the plant so that they can carry out photosynthesis and survive, as we'll talk about in just a moment. But, as you would imagine, when they open that stomata, they lose water. And so they need to have the stomata open to take in CO2, but when it's open, they lose water, and water is very likely a limiting factor depending on where the plant is found. And so it's not something we talk about in this particular class, but as we look across the plant world, we see these fascinating adaptations. If we look at plants that are found in the desert, they have special ways of carrying out photosynthesis and special characteristics associated with these stomata that allow them to carry out photosynthesis in very dry conditions. In fact, to a large extent, that's why we have the trees that we have here in central Kentucky. We have deciduous trees, these trees that have leaves during the growing season and lose their leaves in the winter. And if you look at the leaf of a oak tree or a maple tree or a sycamore, or any of the trees that you see outside, except for evergreen trees, then you see these big, flat, broad leaves. And if you looked at each leaf, you would see hundreds or thousands of little stomata found on each leaf. And each of those stomata allows CO2 to come in and allows water to go out. And so the end of this, the take-home message here is that these plants can only live in areas where there's a sufficient amount of water. We get a reasonable amount of rain here in central Kentucky. We get 40 to perhaps 45 inches per year, which is not so bad. Less than certainly a tropical rainforest, but much more than you'd have in a desert. A desert would have less than 10 inches of rain a year, or a grassland would have 15 inches of rain. And so those are areas where these trees could not survive. It's amazing to think about, but a really big tree, now I don't mean just a moderate-sized tree, but a huge tree that you might find, maybe a couple of the trees found down in the semicircle there in front of Hughes, can release a tremendous amount of water each day during the summer. So when the sun comes up, the stomata are closed, and then say by 10 o'clock or so, the sun's shining down on the trees and these stomata open. They start taking CO2 and they start to lose water. And so one of these big trees can release as much as 100 gallons of water a day through these stomata. Isn't that amazing? That's 100 milk jugs every single day can be released by a really, really large tree during the heat of the summer. And so it's very important then that the trees have, have lots of water that they can take in. And so you realize it doesn't rain every day during the summer, but these trees have roots that are tremendously extensive, and so they're continually sucking up the water from the roots and it's going all the way up the tree to the leaves and is leaving via those stomata. It's just a really interesting aspect of plant physiology. So we want to move on then and talk about what plants do with this CO2 that comes in through these stomata. So photosynthesis then occurs in these organelles that are found in plants referred to as chloroplast. Chloroplast is shown in this on the figure here on our screen, and we want to talk about a couple of the different parts of the chloroplast. The first part that we want to mention is referred to as the stroma, shown here. The stroma is the liquid fluid, the fairly thick fluid that's found inside the chloroplast. And so this whole thing that we see here is the chloroplast of a plant cell. These are arranged into the leaves of plant cells. In some cases, these can be moved from one place to another, depending on the plant that we're talking about. And these are very small. You can see them underneath a light microscope, like you would use in a laboratory, say the laboratories that we have at Asbury. But they are small. They are microscopic structures. So we now want to talk about something even smaller. Inside the chloroplast, we have these structures referred to as thylakoids. Each of these structures that to me looks sort of like a gold coin, a pretty thick gold coin. Now remember, they're cut in half right here, or maybe 25% of them are cut off over on this side of the diagram. But each of those represents a thylakoid. A thylakoid is a disc-like structure, and a whole stack of thylakoids is referred to as a granum, as labeled here. So the stack is a granum, and each individual one is a thylakoid. And so if you are able to look very closely at the surface of that thylakoid, you would see that the thylakoid surface is covered with chlorophyll pigment molecules. And so it's actually the thylakoid that's responsible for absorbing light energy and allowing photosynthesis to occur. Everything that we talk about now pertaining to photosynthesis is going to occur here in those thylakoids. So photosynthesis takes place in two different steps. The first step is referred to as the light reactions. This is the step that occurs in the thylakoids, and the second step is going to occur out in the stroma itself. And so we'll start with the light reactions. The light reactions are so named because they require light, and what happens is that an electron becomes excited. Light energy shines down onto the leaf, and specifically it hits the chloroplast. The chloroplast has receptors for light, and the energy from that light is used to excite an electron. And so energy is imparted to this one electron. And so now the question becomes, what happens to that electron? The very first step is to gather the energy and use that energy to transfer the energy to an electron. So we have one energized electron at this point. And here's what happens to that electron. So the electron, let's look at this diagram here, which is a lot simpler than it actually appears. So the excited electron is found right here. The excited electron enters, where I'm pointing right here, enters into this electron transport chain. And so you remember electron transport chains from chapter 4 in our discussion of aerobic respiration. In this case, the electron transport chain is doing basically the same thing. We have three, in this case, receptors for electrons. 1, 2, 3, as you can see on the screen. And as that electron that has lots of energy jumps from this acceptor to this acceptor to this acceptor, it gives off energy. And then so as a review, you can ask yourself, what happened in aerobic respiration when that energized electron went from one carrier to another carrier to another carrier in the electron transport chain? And you recall, no doubt, that what happened was that hydrogen ions were transported across the membrane. And so that's exactly what happens here. So every time this energized electron, and remember that the electron is energized because of step 1 here, light energy comes to chlorophyll, the electron is energized, and that electron then feeds into the electron transport chain. In this case, hydrogen ions, just like in aerobic respiration, are transported from the outside, or at least the transport is the same as aerobic respiration. Hydrogen ions are transported from the stroma into the thylakoid. And so hydrogen ions build up in the thylakoid. Every time a single electron goes from here, where I'm pointing on this first acceptor, to here on the second acceptor, one hydrogen atom goes from out here in the stroma to in here inside the thylakoid. Same thing happens at the second acceptor, same thing happens at the third acceptor. And so now we have that electron that's made its way all the way down to this last acceptor on the electron transport chain, and at this point you can ask, where does it go? And here's where it goes. It goes to NADP+. So NADP plus is just like NAD plus in aerobic respiration. And so NADP plus acts as a little taxi for electrons, just like NAD plus did in the case of aerobic respiration. And those electrons then are going to be taken over to the Calvin cycle, the last part of photosynthesis. And so when the hydrogen ion is, okay, perhaps this is a little bit confusing. So here we have the electron gets off the end of the electron transport chain, and it attaches to NADP plus. And actually two of them go through. One goes through and attaches to NADP plus, another goes through and attaches to NADP plus. And when that happens, one hydrogen ion is also added to NADP plus to make NADPH. So what you should remember is this, that NADPH is a molecule that's carrying 2 electrons, or you don't even have to know 2, just remember that it's carrying electrons. That's the purpose of NADPH in photosynthesis, just like the purpose of NAD plus in aerobic respiration was to carry electrons. Alright, and so that really tells us most of what we need to know here. If you understand that, you understand most of photosynthesis. So a couple other details that we want to cover here that are important. One is that we're all aware of the fact that oxygen is released as a result of photosynthesis, and so that allows life on Earth to persist as we know it. With every breath you have to take in oxygen. And so where does that oxygen come from? And interestingly, all of that oxygen comes from what we see occurring right here. So the electrons have to, they are excited in the chlorophyll here, and then they feed into the electron transport chain. And so every time an excited electron leaves, it has to be replaced from somewhere. And you have to ask yourself, where did those electrons come from? Electrons are continually leaving and going down the electron transport chain. They've got to be replaced somehow. And how are they replaced? Here's how. We take water, and water is split. So when water is split, hydrogen ions remain inside the thylakoid membrane here, and oxygen leaves. And so this is the oxygen that's keeping you alive right now. All the oxygen on the face of the Earth, I'm sorry about that, all the oxygen on the face of the Earth comes from this particular part of photosynthesis. So we split water, and we pull those electrons away from water, and we use those to replenish the excited electrons that are leaving from chlorophyll and feeding into the electron transport chain. And so this is where oxygen comes from. Interestingly, oxygen, this doesn't help plants at all. This is just a byproduct of this whole process. Plants need those electrons to feed down the electron transport chain for purposes that we'll see in a moment. But they don't care what happens to oxygen. Once water is split, oxygen just leaves. But you depend on oxygen to remain alive. With every breath, you have to have that oxygen to use in aerobic respiration, as we saw in Chapter 4. Alright, and so everything else here is going to look just like we saw in aerobic respiration, or at least this last step is. So here we have a high concentration of hydrogen ions inside the thylakoid membrane. And what's going to happen here? We already know the answer to that. Once again, we have this molecule ATP synthase that we see in the bottom portion of our screen here, ATP synthase. And ATP synthase makes ATP by allowing hydrogen ions to go from an area of high concentration to low concentration, diffusion in other words. And so hydrogen ions want to go out this ATP synthase molecule. It creates a little tube, and you already know all of this. I'll remind you from Chapter 4. This is the same molecule that we saw in cellular respiration. It's just it's working in a different process in a whole different organism, a plant, instead of an animal at this point. So hydrogen ions flow through ATP synthase, and as they flow through, ATP is created. So that finishes our discussion of the light reactions. And so let's briefly review what we gained. We won't go back through the whole process, but what do we get from this whole process? And the answer is we gained two things. We got NADPH, and NADPH is carrying electrons. And so that's what we have there. We have electrons in the form of NADPH, and then we also have ATP. And so those two things are going to move on to the next step of photosynthesis, called the Calvin cycle, and be used to complete this process. Alright, and so the Calvin cycle then takes place independently of light. It's also referred to as the light independent reactions. and it takes place out in the stroma. No light is occurred for it to take place at all. And here's what happens in a nutshell. So this is a nice diagram from our text that summarizes everything. Here's NADPH, and of course that's carrying electrons to the Calvin cycle. Here's ATP, that's taking energy to the Calvin cycle. And so electrons and energy are used in the Calvin cycle to take, this is the most important thing perhaps in this whole process, to take CO2, which is feeding in here, and to make sugar. So here's ATP, that provides energy. Here's NADPH, that provides electrons. And here we have CO2 feeding into this cycle. And the combination of those three things, energy, electrons, CO2, makes sugars. So CO2 produces, or provides rather, the building blocks for those sugars, and then the electrons are used to connect the different molecules of the CO2 together, and ATP is used to provide the energy to drive this process and to be stored, actually, interestingly, in those sugars. And then, of course, here we have the spent ATP portions, ADP, adenosine diphosphate, just like we saw in Chapter 4. We had a whole lecture just on that in Chapter 4, as you remember. And here we have the spent NADPH, which is now NADP+. And so it's the same molecule, it just doesn't have any electrons. These go back to the thylakoids, and in the light reactions, they get more electrons and they get energy, and they go back to the Calvin cycle. And so this goes around and around and around, as we see here, as long as a couple things are occurring. As long as light is coming in, as long as water is present to provide these electrons and to allow oxygen to be released, and as long as CO2 is present. So that takes us back to our discussion of stomata. This whole process is not going to occur unless those stomata are open. So I think we should talk just a bit more at this point about the importance of this. And so this is referred to as carbon fixation. Carbon is coming in to this chloroplast, and the plant is taking CO2 from the air and making it into a sugar. These sugars are then used to both provide energy for the plant and structural materials for the plant. And so this is really an amazing concept to think about. But imagine a redwood out in California. Those trees can be as wider than a school bus at the base of the trunk, and 300 feet in height. A massive, massive living organism, some of the biggest on the face of the earth, and thousands upon thousands of pounds of mass of wood and leaves and roots found in those trees, and they're hundreds of years old. And so remember that every single bit of the mass of that tree came from CO2. So that's what plants do. Plants pull in CO2 from the atmosphere, and they use it as building blocks to make everything about them. The bark, the trunk, the leaves, the fruit. If you're growing a garden this year, remember your tomato plants are pulling in CO2 from the atmosphere, and they're using that to build the leaves of the plant. They're using it to build the tomatoes and so forth. Now, yes, they do need fertilizer. That's a limiting factor in allowing them to carry out these various processes. But that's not what the leaves and the fruit and the mass of the plant is made of. It's all made of carbon-based molecules that come from CO2. This is very important in that idea of the carbon cycle, as we talked about in the last lecture. So this means that if we look at the face of the earth, then there is a tremendous amount of carbon tied up in forest that we see on the earth. That carbon is found in the leaves and especially the woody portions of trees. So if we were to cut those trees down and burn them, we release that carbon back up into the atmosphere.